Other people study the human. We do a bit of work around the virus, and I'm interested in the mosquito. I grew up in Flint, Michigan. So Michigan is a state in America that looks like a mitten, and it's surrounded by lakes and touches Canada, so it's quite cold in the winter, and Flint's about right there. And so I used to get outside the city a bit because I really liked animals and biology and spend time at my grandmother's farm. And so it was, I think my early interests grew out of spending time outside and having to get beyond the city to do that. I'm really interested in how the insect transmits disease and its role to play in human disease transmission. Trying to understand a bit about when a virus, dengue virus, arrives in mosquito saliva. So a mosquito takes a blood meal that is infected, so if it bites a human who's got dengue virus, it takes time for the virus to move through the mosquito's body, reinfect the salivary glands and be put into the saliva. So that time window is really important. That delay is what determines how much disease a single mosquito can cause in its lifetime. We're getting in shipments from the field every week or so. There's a bit of a rolling process to check their species identification and then get them blood fed in the lab and collect eggs from them. We're interested in looking at genetic diversity and dengue susceptibility from wild populations. So we want to compare across quite diverse genetic lines that we're bringing in from the field. My group has discovered a way to study mosquito saliva and the arrival of virus in the saliva. And we can check every 2 days for a single female and ask, is the virus here yet? Is it here yet? And also ask how much virus is present. So that lets us have a real tight relationship between that single mosquito's ability to transmit disease and her genes.
These ones still need to be screened to check that they're actually the species that we're after.
We've got people in the lab who just have a natural skill and a green thumb for mosquitoes. So they're really important contributors to the group because they have that sense for what do the mosquitoes need. And it might mean varying the food, it might mean there's too much light coming into their cage, or they don't like these cages, they're too small, too big. Having a real sense or feeling for the organism. We've found for a really strong virus some data suggesting that the virus arrives very early, 5 days, which is much earlier than was predicted. And we've also shown that virus sort of disappears out of the saliva in old age mosquitoes. And again, that's something that wasn't really expected. And all of that will contribute to the nature of dengue transmission in the field.
I don't know.
And my group runs really huge experiments where everyone has to come together and help each other. And then sometimes they fail. And then we have to do them again. So for us, it's around, the challenge is often around execution and scale and trying to take a question from something we're really interested in to get it to work. I think at a lot of our experiments we have false starts and so we learn something each time and we get closer to being successful. And so I'm used to that and my students are used to that now. It's part of the process of how we do science and I'm okay with that.